The Parkinson's podcast is brought to you by the Davis Phinney Foundation and brings you the stories, wisdom, and expertise of people living with Parkinson's, care partners, and Parkinson's professionals. In this episode of the Parkinson's podcast, Dr. Arash Fazl discusses the role of the protein Alpha-Synuclein and how this protein is involved in new diagnostic tests for Parkinson's. Dr. Fazl also shares how these new tests may contribute to additional exciting developments in the future. This episode is part 2 of a 2-part series about Parkinson's diagnosis. In the first episode, Dr. Fazl describes the challenges involved in diagnosing Parkinson's. This episode picks up in the middle of our conversation with Dr. Fazl, right where we ended the first episode. This 2-part series is sponsored by CND Life Sciences, home of the SYN-1 test, the first commercially available skin-based test to help clinicians diagnose Parkinson's disease and related disorders.
In recent years, there's been a lot of big news about developments of biomarker tests and then the SYN-1, the SYN-TAP tests. And I'm just wondering if you can explain a little bit about progress in diagnostic testing that relates to alpha-synuclein. And I guess the first part is to say, what is alpha-synuclein and what is its relationship to Parkinson's?
Yes. If you look at the history of Parkinson's disease, first they discovered the role of dopamine. They realized that somehow dopamine goes down in the brains of patients with Parkinson's disease. And they thought, okay, we solved the problem and we just give dopamine to patients and we basically treat this situation. And then they realized, no, there is something else going on. And it seems that the dopamine is a innocent bystander that gets hit in this process of the disease. Then they started noticing this protein aggregates under the microscope. They started seeing these clumps of abnormal protein in the neurons or in the dendritic tree or in the tails, the axons of these cells in the brain and also in the periphery. They said so, and they were seeing this very commonly. It was found in the majority of the autopsies of Parkinson patients. So even though the dopamine story was basically making the most impact on the treatment of Parkinson's disease, there was pathologically this finding that was very ubiquitous in a lot of patients with Parkinson's disease. So they finally found that these aggregates seem to be the clumps of a particular protein called alpha-synuclein. And then they said, so what is this protein doing? Where do we see this alpha-synuclein? And they found it very commonly distributed in the brain especially. And it's not just something that humans have. It's a rather old protein, meaning that in evolution it has been with us going back, not only the mammals, it can be even found in the bony fish, some versions of it. So it is something quite important when the scientists basically reason that it must be doing something very fundamental, that we have kept it in all these stages of evolution from fish to mankind. But they think that this is involved in the working of the synaptic vesicles, which are these little blobs that gather neurotransmitters, proteins that take messages from one neuron to the other one. They are packaged, and in the wall of this blob of packaging, this alpha-synuclein plays an important role to basically have these synaptic vesicles transmit properly, go run to the end of the neuron and gets released and release their contents to the next cell. So they are playing an important role, and if they are just remaining normal, nobody would have found that they are involved in Parkinson. But some process apparently makes them go rogue, and they clump together and form aggregates of 3-4 more single monomers. And that form apparently is toxic to the cell. We exactly don't know why this happens and when they happen, how it exerts that toxicity, but they think that it increases the inflammation in the cell and somehow it injures the cells and kills the cell, that aggregate. So that's the story of alpha-synuclein. Now, when we find that, of course, the next question, wow, so if this is so integral to Parkinson's disease, maybe we can use it to detect Parkinson's disease. Go look for alpha-synuclein. And if we find this aggregated, toxic, pathological form of alpha-synuclein, then that person has Parkinson's disease. Again, we still haven't really honed in that, is this a correlation or causation? We still don't know. Just they happen to be both present, but not really part of the disease process or not. But bottom line is that if something happens in such high prevalence in Parkinson patients, detecting it can help us basically diagnose Parkinson. And that is the basis of two new tests that now we have in the past few years going and detecting directly this alpha-synuclein and not relying on all those indirect ways of diagnosing Parkinson's disease.
Let me just see if I understood this. So alpha-synuclein is a protein that is in our bodies, and it has been in the human body for a very long time, and it's also in a lot of other animals. So it's important because it's been around a while. We understand that alpha-synuclein is involved somehow with the transportation of certain parts of the neuroanatomy and the neurobiology, maybe. I'm not sure how to put that, but they somehow involve movement of something from one place to another in the brain. And somehow, we're not sure how, these proteins can clump up on themselves, and they can thereafter damage the neurons, or at least correlated with damage to the neurons.
Yes. Somehow, when the single proteins clump together, they become toxic, they injure the cell, they kill the cell. And one theory is that they spread and turn the good alpha-synuclein in other cells into bad clumped aggregates, and the cycle continues. And that's how the disease progresses.
And so these aggregates are findable by some new tests. And those tests, maybe you could say a little bit more about those tests and how they work. As I understand, these tests are the SYN1 test and the SYN-TAP test. Those are the names, as I understand. Is that right?
Yes, yes. So through the support of Michael J. Fox Foundation, a lot of researchers actually combined forces, and they basically evolved their technique to find the small amount of alpha-synuclein that can be found in or around the brain. Now, I just alluded to one interesting property of these bad protein, alpha-synuclein aggregates, that apparently can make the good protein evolve into the bad protein. They're basically like a zombie process. They bite the good proteins and they turn them into bad proteins. So they used a similar trick to augment, to basically increase the amount of the bad protein in their sample, because the amount that you get in the cerebrospinal fluid is so small that your devices cannot detect that. But if we introduce some alpha-synuclein into the mix, they aggregate and that seed amplifies. It's called seed amplification assay, and it basically increases so much that then you can detect it. And they now can detect alpha-synuclein in the cerebrospinal fluid. And that's the basis of SYN-TAP. It initially was used in research, but now it's clinically available. Another group of scientists, they were looking at the expression of alpha-synuclein pathologically in other tissues of the body. And eventually, in some pioneering work coming from Harvard University, they showed that you can find this particular pathological version of alpha-synuclein in the nerves of the skin, in the autonomic nerves of the skin. And that is the basis of the SYN1 test. For this test, you just need a small sample of the skin. We send it to the lab, and they look for the nerves of the skin and also alpha-synuclein. If it exists in the same location as the nerves, they can say that these nerves are affected by phosphorylated alpha-synuclein. Hence, it is positive, and you most likely have Parkinson.
Okay, so these two tests that you just described, SYN-TAP and SYN1, are doing similar things to detect alpha-synuclein that is misfolded or that misfolds.
Yes, exactly. It's pathological, and it is not just normal synuclein. They find the pathological forms of alpha-synuclein.
Okay, so these tests are done either with spinal fluid or with skin, and the spinal fluid is obviously coming from the spine, but with the skin test, there's different places, as I understand it, right? I think you might have alluded to that, but I think it's true in any case that the tests can be done in different places.
Yes. So many labs looked at how we can find alpha-synuclein in accessible tissues of the body. And skin, of course, is the most available, accessible place in the body. And when they found it, then they should basically, in order to give it to the doctors and be used in the clinic, they should say how accurate this test is, how sensitive it is, and how specific it is. How sensitive is means that if you have Parkinson's disease, how many times this test detects you as positive. In order to do that, one test that they did was to find out what's the relationship between number of samples we get from the body and the accuracy of our test. Of course, we want to take the minimum amount that gives us the best answer. The numbers that I remember seeing was that if you just got one location, one sample from the skin, you might detect in about 50-something% of patients. If you do two tests, two sites, you go up to 70-something%. And with three locations, you reach about 92-95%. And if you do 4 or 5 locations, you have already exhausted most of the accuracy. You don't go much higher. Maybe you add a few more percentages. And they have found that the 3 better locations are the back of the neck, around C3, C7 area, to the side of the neck, 10 centimeter above the knee, so lower thigh, and 10 centimeters above the ankle, lower leg. And with these 3 locations, the accuracy of the test for detecting Parkinsonism is about 90-93%, which is a very high number, meaning we really can, change our diagnosis based on an accurate test like this. It is no longer a circumstantial test. It really tells us something. So that's one thing we like about the test of SYN1, that is very highly accurate. Number 2 is that it's very easy to do, and it doesn't need expertise of doing a spinal tap. Definitely, I think spine is a more sensitive sample. It can give us a lot of information about the brain. And in certain circumstances, probably we would love to have the spinal fluid. But at least for the usual clinical testing, this skin biopsy is a very fast test. It can be done by a nurse, doesn't need a doctor, and very quick, within 10 minutes, you collect those, doesn't need a stitch. You just put a Band-Aid on those 3 sites. Very rarely it can have a local infection or bleeding. I have done, maybe hundreds of these and really have not had any bad experiences with them. So easy test and very high accuracy. So we really like it. And in all those gray zones where I, as a doctor, am not sure about the diagnosis, or my patient is not sure and has gone from one doctor to the other, asking for second and third opinion, doing this test not only gives me confidence, but also tells my patient that the diagnosis is correct and focuses their attention now on treatment and, the next steps in their journey.
So this is really exciting because I think what you're describing is that there are options for people who potentially are living with Parkinson's. There are multiple ways to get clarification about the diagnosis. And now there's multiple tests rather than really one kind of exam in the past, which was the DAT scan. Now there are 3 tests that can help us clarify the diagnosis. I have 2 questions about the SYN1 and the SYN-TAP test. And the first one is, are these tests certainly showing that somebody has Parkinson's disease rather than some other type of Parkinsonism, or is there some still gray area?
Yes. So let's dive deeper on that front. As a clinician, first you ask yourself, do I see Parkinsonism? Those 3 physical exam signs that puts it in the area of Parkinson's disease and its related disorder: slowness of the movements, rigidity, and tremor. Then you ask yourself, what kind of Parkinsonism am I dealing with? Am I dealing with the classic Parkinson's disease? Am I dealing with Lewy body dementia? Am I dealing with MSA? Am I dealing with PSP, vascular Parkinsonism, drug-induced, et cetera, et cetera, et cetera? So now that I'm in this big umbrella, which one is it? And a lot of times the history helps you. If your patient hasn't taken enough these medications, drug-induced is out. If they don't have significant cognitive difficulty, Lewy body is out. If they, have a lot of these mini strokes in the MRI of the brain, the vascular Parkinsonism is out. So you can rule out a lot of them easily, but sometimes, especially between PSP, MSA, and Parkinson's disease, between these three, you find yourself in not the majority of cases, but in a minority of patients, you find yourself guessing which one of these three you're dealing with. And then DAT scan and skin biopsy can come to your rescue. Remember that I said DAT scan is a less selective test. It turns positive in all of MSA, PSP, PD, and Lewy body. So if you were thinking about PSP, then your DAT scan would be positive. But PSP is not one of those diseases that is based on alpha-synuclein. So your skin biopsy should be normal, should not show any abnormality. So sometimes we use that in the clinic, both DAT scan and skin biopsy, if we think somebody has PSP. The DAT scan should be abnormal, the skin biopsy should be normal. And that tells us that maybe if it fits our clinical diagnosis and our history and a lot of other things, then we think that, okay, the tests are pointing in the direction of PSP. So that's one way of using skin biopsy and DAT scan in tandem to hone in on accurate diagnosis. And then the plot thickens even further when now you're looking at very similar pathologies of MSA, Lewy body, dementia, and Parkinson's disease. Now, these three, all we expect, turn your DAT scan and skin biopsy abnormal. So how do you tell them apart? Which is one of the areas that a clinician like me sometimes finds ourselves scratching our head and thinking, well, it has overlaps of like the blood pressure is dropping. Is this MSA or is it PD? I'm not going to get to the diagnostic intricacies of this, but it's not sometimes easy for a doctor to tell PD and MSA apart, especially in early years of the disease. And the group that introduced the SYN1 test, they also have done small studies, not very big studies, but small studies that showed there might be some difference in the pattern this test turns positive in MSA versus PD versus Lewy body dementia. And so I have actually used that knowledge, and in the cases that I was in doubt, even reached out to the CND labs who have introduced SYN1 test and said, well, I think clinically my patient has MSA. What does this pattern of positivity in the skin test tell me? Do you think also this more points toward MSA? And they basically have helped me narrow down my diagnosis in a positive test. In a nutshell, in MSA, we have a higher chance of all those three locations in the neck, in the thigh, and in the leg turning heavily positive. Whereas in Parkinson's disease, usually either the neck and the thigh at most turn positive. Usually you have more proximal, toward the head distribution of alpha-synuclein, whereas MSA seems to be all over the body. Also, Lewy body seems to be all over the body. And then the neuropathy in the test also tells you other things about the nerves in the body, how dense the nerves are in that skin sample in each location. And the nerve density seems to have dropped in Parkinson's disease. You have a peripheral neuropathy, whereas in MSA, you might not have that. And so that is another way that we can tell PSP and MSA apart, only using this test. But of course, there are other things that helps us. So that is one of our circumstantial evidence to tell them apart. So this test actually has started already proving even more useful than just detection of Parkinsonism, but maybe tell apart the different subtypes of Parkinsonism, PD versus MSA.
This is very helpful because this can be a scary process for folks. And that's the point of these tests really, is to help us gain more information to be more certain about what we're dealing with. So I said I had two questions. And the last question is it possible for someone with Parkinson's to not have an abnormal alpha-synuclein indication in a test?
Yes. We have seen that some patients who have clear Parkinson in the exam and have good response to levodopa, the two measures that we clinically say this is highly probable clinical diagnosis of Parkinson. They don't have positive skin test. Some of them are really, true negatives, and some of them can be missed by the test. Maybe it's one of those few percentages that if we have sampled more sites in the body, we might have found it. So some are missed because of that. But those are just a few percentages. But we are also learning that Parkinson's disease is not a homogeneous block. It's not a monolithic entity. There are subtypes, there are variations, there are genetic variants of Parkinson's disease. And one of those genetic variants that is highly causing Parkinson's disease, called LRRK2, LRRK2 mutation, actually has less chance of causing a positive skin biopsy. It's a lot of times indistinguishable from normal, non-genetic versions of Parkinson's disease, very similar to the classic Parkinson presentation, but they are famous for having much less positivity in the skin test. So those are the two examples I can think of. And then there seems to be some authorities, some clinician scientists are saying that we have two origins of Parkinson process. One starts in the body and makes it to the brain. The other one starts in the brain and then makes it to the body. So you can imagine the second type that is rather less common, maybe 10% of Parkinson patients, the process starts as an inflammation or something in the brain. It hasn't affected the body, so it is not found in the peripheral nerves. That might be negative in the skin biopsy. So those are the two. But again, this is the edge of discovery. We are not 100% sure. So we have to be aware of the limitations of the skin test as well.
Thanks for sharing that extra information. And I appreciate that this is, we're on the cutting edge here. And that's what's so exciting about this, as you said at the beginning, that there's a lot of things moving around right now in terms of our understanding of Parkinson's. And so I just want to draw attention to one little wrinkle in this, which is that we didn't talk about genetic testing, but I think what you just said suggests that there is a role for genetic testing in this whole process of diagnosing Parkinson's. In particular, if one were to have a pretty clear clinical presentation, but not a clear representation of abnormal alpha-synuclein in an alpha-synuclein test, you might be well served to consider a genetic test to see if LRRK2 is present, because that might be further evidence suggesting that Parkinson's disease would be an appropriate, all those things might be compatible with a Parkinson's diagnosis.
Exactly. And there are more reasons to consider genetic testing. One is everything points to Parkinson's disease, the skin biopsy is negative, so you suspect the genetic form. That's one reason to go and figure it out. The other one is when your patient has a very strong family history of Parkinson's disease and they're worried about their children. Is my child, who is an adult and going to have children of their own, are they going to basically pass this gene? So genetic testing becomes important for them. Ashkenazi Jewish ancestries has a high percentage of that. So there's a lot of reasons to consider genetic testing. Right now, today, we don't have a medication specifically for one of these genetic forms. So it doesn't change our usual approach, but that landscape might be changing. There are clinical trials underway for LRRK2, for GBA, to say at least two of them, if not more, that we are looking at specific treatments for this genetic form. Then this genetic test becomes way more important. Then everybody should get those genetic tests. And other reasons that I just on top of my head I'm throwing here is basically people we are sending for deep brain stimulation. We are learning that some genetic variants might not do very well in DBS. So we have started following some research that we want to do genetic testing for all the patients we send to deep brain stimulation. And so there are more and more reasons to basically think it is not a monolithic diagnosis. Different versions of it, different genetic variants behave differently, and we have to tailor our management and our treatment to specific patients. So yes, there is a role for genetic testing, and it goes beyond just the skin biopsy.
And it also will probably increase. And a critical point of what you said is that the utility of genetic testing is likely to increase over time.
Oh, yes.
So that is another thing to consider. It might inform your ability to participate in clinical trials, which can be especially interesting when you're recently diagnosed and you might not necessarily need medication to live your life without any hindrance, potentially. Or even if you do need to take some medication, it might still be useful in helping you find a clinical trial. So, Dr. Fazl, I really appreciate all your time. I guess I want to ask one last question, which is just, what do you hope for next in terms of diagnosis?
We are like kids in Christmas. When we are given a gift, we always want more. We always wish for more. So any of these tools that come out from science and help us in the clinic opens new doors and new questions. We start asking, so how can we basically use these tests to predict different subtypes of Parkinson? Are there different subtypes? How can we predict how one person progresses? How is this test telling us about the response to medication? How can we tailor medications to our patients? And I think they probably will serve more and more utility in the clinical trials. Now we have basically limited tools to say if our disease-modifying agents, that they are starting to become more and more common in the clinical trials, meaning that the pharmaceuticals are throwing their might of research and money behind innovation in trying to slow down the progression of the disease. We are still relying on physical examination to predict that the disease has slowed down. It would be way more accurate if we could find a good biomarker, a good test to help us say that the disease has slowed down. So a lot of these areas, I think, are ripe for discovery. And I particularly highlight one. We have not yet, unfortunately, found a substance, a chemical that slows down progression of Parkinson's disease. But we might not be that far. And if that happens, we really need a test to detect Parkinson's disease as early as possible, because it's a very slow moving process. Imagine how we can change the life of our patients if we had detected them even before their physical signs appear. And some of these tests are actually doing that. We can detect some of these patients before their movement is affected, not rely on our physical examination. Imagine we start slowing that process at that stage. Imagine how better the quality of life of these patients will be. So I cannot wait for that day that we can detect it early and we have found substances that can slow it down. Those two go hand in hand in making the, starting the golden age of Parkinson treatment. Hopefully we are not that far.
Well, it sounds like the recent developments in diagnostic testing are pushing us along that road pretty substantially. So that's exciting news indeed. And I appreciate you sharing your insights about these developments with us and our audience. And, I really do appreciate your time. So thank you very much. And I'll look forward to talking with you again sometime very, very soon.
Thank you for listening to this episode of the Parkinson's Podcast. To hear the first part of our conversation with Dr. Fazl, which covers the diagnostic process more broadly, you can find the link to that episode in the description below. For more information about the Davis-Phinney Foundation and to learn about educational offerings and community events for people affected by Parkinson's, please visit davisfinneyfoundation.org or dpf.org. This podcast includes information about Parkinson's and insights from our Parkinson's community. It is not intended as a substitute for treatment advice from your own medical providers.